Welcome to the first of 2 lectures about the central nervous system. So the CNS consists of the brain and the spinal cord. So exercise 17 focuses on the brain part of the central nervous system, and exercise 19 then focuses on the spinal cord portion of the central nervous system. We're also going to talk about the cranial and spinal nerves in each of these lectures, even though they're actually part of the peripheral nervous system. So when it comes to the brain, there are kind of 4 major regions of the brain, and they all kind of carry out different functions for us. So the largest portion of the brain is known as what is the cerebrum, or the cerebral hemispheres. This is really our cognitive brain, our high-level brain that allows us to interact with the world, to have thoughts and feelings, etc., etc. Interior in the brain is what is known as the Diencephalon, which is a relay station which carries out some homeostasis functions, so a very important part of the brain as well. And then connecting those higher centers then to the spinal cord is the brainstem. Midbrain here, pons here, medulla oblongata here. And you can think of the brainstem as, sometimes it's referred to as our quote-unquote lizard brain, because it's a very primitive part of our brain. It carries out things like keeping our heart going and our respiratory rates normal. It does connect the spinal cord to the brain and the brain to the spinal cord, but we'd be really in big trouble if we lost our brainstem. And then in the back portion here, we have the cerebellum. So this is a kind of motor function, or a motor part of our brain. It's also where we process a good chunk of our kind of balance information. Typically people who have damage to their cerebellum they walk like drunken people, basically. So if we focus on the cerebrum first, the higher brain functions, you'll notice that the brain itself is not smooth. It has all of these kind of ridges and grooves in it. And now our brain will be kind of more deeply ridged and grooved than the sheep brains that you're actually going to get a chance to dissect, because we have more neurons. We have more cognitive power than the sheep. The purpose of the ridges and grooves is to create more surface area to accommodate more neurons. More neurons, more cognitive power. That's why our brain is more ridged and grooved than the sheep brain. So a ridge is known as a gyrus, or gyri, plural. The shallower kind of grooves is a sulcus, singular, or sulci, plural. And then these really, really deep grooves are then known as fissures. So there are 2 hemispheres, a right and left hemisphere. Connecting them is the longitudinal fissure. There are 5 lobes of the cerebrum, mostly named for the bones that overlay them. So you have the frontal lobe in the front, parietal lobes on the sides, like top sides, temporal lobes, sides, occipital lobe in the back. And then there's this deep interior lobe called the insula. And it's the only one that's not named for a bone. If we look at the kind of divisions, what separates the lobes, you'll see that the central sulcus divides the frontal from the parietal lobes. The lateral sulcus divides the parietal lobes from the temporal lobes on each side of the brain. And then in the back, you have a parieto-occipital sulcus that's going to divide the parietal from the occipital lobe. So let's take a look, right? So here's a gyrus. You can see shallow groove is a sulcus, deeper groove is a fissure. So it's all about making sure that we have more surface area for more gray matter, more neuron cell bodies, in order to have more cognitive power. So frontal lobe here, parietal lobe here. The frontal and parietal lobes are separated by the central sulcus, and so the 2 gyri to either side of the central sulcus is the postcentral gyrus on the parietal lobe and the precentral gyrus on the frontal lobe. The lateral sulcus separates the parietal lobe from the temporal lobe, and then the let's see, can you see it? Yes. Parieto-occipital sulcus separates the occipital lobe from the parietal lobe. And then here again you can see cerebellum in the back and parts of the brainstem. So when we talk about the cerebrum, the outermost portion of the cerebrum is known as the cerebral cortex. And really you can think of the cerebral cortex as basically the CPU for your brain. It's where all of the cell bodies of all the neurons are, and it's really what is responsible for our ability to sense things and to interact with the world. So it's divvied up into very discrete kind of what we call domains for very specific motor and sensory functions. So the parts of your brain that light up on a scan when you're using your eyeballs is the occipital lobe back here. When you're hearing things, it's this portion kind of mid-side that lights up. Same thing here with speaking. When it comes to those kind of higher level functions, more consciousness, cognitive thinking, then it's not just a single part of your brain that lights up. It's pretty much the entire cerebral cortex. And so all of the high level thought that we can do is going to involve not just parts of the cerebral cortex, but pretty much kind of all of it working, not just as their discrete parts, but kind of the whole is greater than the sum of the parts. There is also what we call contralateral control when it comes to the cerebral cortex, which is that the left hemisphere of the cerebral cortex controls the right side of the body, and the right hemisphere of the cerebrum controls the left side of the body. So the sensory nerves coming up to the brain and the motor nerves coming down from the brain, they will have to cross over somewhere, either in the brainstem or the spinal cord, because it is true that right brain controls left body, left brain controls right body. Now, you may have heard this idea that left-brained people are really analytical and logical, and right-brained people are really artsy and creative. And while it is true that there is some what we call lateralization between the two hemispheres, meaning kind of we divide up some of the cognitive labor between right brain and left brain, the idea that you are one or the other, that there's this clear demarcation of left brain logic, right brain art, is kind of bunk. It's nonsense. Now, for most of us, our verbal language centers will be on the left side of the brain, and our kind of nonverbal language centers will be on the right side of the brain. But that's kind of a general rule of thumb, and it's not quite as clear cut as that whole right brain, left brain kind of mythos that you may have heard before. So if we look at the different parts of the cortex, we have what we call sensory cortices, motor cortices, and then it's really kind of an association cortex. So each of our senses gets a primary cortex and an association cortex. So a primary cortex is what actually allows us to see, to hear, to smell, whatever the case may be. The association cortex then helps us interpret that. So in order for you to recognize that you are looking at a picture or that you are looking at a computer screen, the fact that you can see the computer screen is a result of a functioning primary cortex. The fact that you can understand what you're looking at is a function of the association cortex. And then something very similar happens with our motor function as well. The primary motor cortex is what actually controls all of our voluntary movement, allows us to walk and talk and move. But the premotor cortex helps us kind of coordinate that. So if you were to move reflexively, that means your primary motor cortex is still functioning. But if you couldn't figure out how to move in order to respond to something, it might not mean that your primary motor cortex is not functioning. It might mean that your premotor cortex is not functioning, because you can't like make sense of it. You can't plan it. And then we also, way in the front of the frontal lobe, have this really, really important cortex called the prefrontal cortex. And it is where you are you. It's where we are able to reason. It's where our intellect comes from. It's where all of our kind of rational thought comes from. And what's really interesting about the prefrontal cortex is that it's the very last part of your brain to develop properly. It does not fully develop until you're in your kind of early to mid 20s. And so if you've ever made like a perhaps unwise decision as a teenager, in part that is because that reasoning ability is not quite fully formed yet. We also have a couple of really important areas that are really well developed in our brains because they relate to speech. So we have a motor area on the frontal lobe called Broca's area, which is what allows us to kind of shape our tongue and move our tongue in order to make words. And then on the back, well, more towards the parietal lobe, we have an area called Wernicke's area, which activates in order for us to understand speech and language. So let's take a look at the different cortices. So if we look at the motor, let's look at the motor cortices first, mostly because there are less of them. So here on the prefrontal or the precentral gyrus is the primary motor cortex. Just anterior to that is then the premotor cortex. So if the primary motor cortex is damaged, you'll be paralyzed. If the premotor cortex is damaged, you'll still be able to move, you just won't be able to kind of plan on moving, if that distinction is clear to you. If we look way up front here, very front of the prefrontal cortex, this is our working memory. This is our executive area. This is our object recall. It's our thinking and our intellect and our cognition way up here in the prefrontal cortex. You'll also then see this Broca's area here for our ability to speak. If we then look at pretty much every lobe except the frontal lobe, that is where we will find our kind of sensory cortices. So on the postcentral sulcus is our primary somatosensory cortex. Somatic sensation is our like touch and temperature and pain sensation. Much of the rest of the parietal lobe is then an association area for our somatosensory touch senses. On the back, on the occipital lobe, we have our primary visual cortex and our visual association area. The auditory cortex and its association area are located here on the temporal lobes. The gustatory cortex, for our ability to taste, is actually on the insular lobe, so kind of at the kind of meeting point. Oh, come back here. Ooh, right here. And then this dashed area here, which spans some of the temporal and some of the parietal lobe, is the Wernicke's area for kind of language understanding. And so again, if your primary auditory cortex were damaged, you couldn't hear. But if you could hear, but what you heard was gobbledygook and not anything that you could understand, that would indicate damage to the auditory association area. So the primary sensory cortices like impart the sense. The association cortices help us interpret it and make sense of whatever sense that we happen to be talking about. If we look internally in the cerebrum, we're looking at primarily white matter, connecting all of these kind of cell bodies of all these different neurons in the cortex. Association fibers carry information within a hemisphere, so maybe from the premotor cortex to the primary motor cortex. Projection tracts are for connecting higher and lower brain centers, so from the cortex to maybe the a diencephalon or a spinal cord, and then commissure tracts connect the two hemispheres, and the most important of the commissures is the corpus callosum. And in fact, the corpus callosum allows such a high degree of synchronization between the right and left hemispheres. That's part of the reason why that whole kind of lateralization, right brain, left brain thing is nonsense, because there's such kind of seamless communication. Now, if we look internally in the cerebrum, you'll see while it is true that most of the internal cerebrum is white matter, there is also a little bit of gray matter as well. It's called the basal nuclei. They help us regulate, particularly like really high order voluntary motor control. People who have Parkinson's disease tend to have damage to their basal nuclei and also parts of their brainstem, and they can't move as kind of smoothly and seamlessly. And then the basal nuclei also play a little bit of a role in our kind of learning and our memory. There are three sets of nuclei that are part of the basal nuclei: the caudate nucleus, the putamen, and the globus pallidus. If we then look at the diencephalon, if we look at the diencephalon externally, we'll be able to see the olfactory bulbs connected to the olfactory tracts. We also might see a little kind of structure called the mammillary body. So our olfactory bulbs and our olfactory tracts are, of course, taking information, olfactory information, smell information from our nose to our brain. And then the mammillary bodies actually link memory and smell. So like if you smell something and it like immediately recalls a memory, part of the brain that is working at that time is the mammillary body. You'll also see the optic nerves and tracts so that we can see, and the nerves and the tracts, they actually cross over, again, right brain, left brain. That cross is known as the optic chiasm. And then, of course, we'll also see the pituitary gland, which is a really important part of the endocrine system. So when you're looking for external diencephalon features, you're really looking at not the dorsal part of the brain, but the ventral part of the brain, at least on the sheep brain. Internally in the diencephalon, the three kind of major areas of the diencephalon are the thalamus, the hypothalamus, and the epithalamus. So the thalamus I like to think of as Grand Central Station, because basically everything comes into it and then everything comes out of it. So all of your sensory data, with the exception of your sense of smell, goes into the thalamus. The thalamus kind of sorts it, packages it, and then sends it onward to its destination, whether that's up to the cerebrum, over to the cerebellum, from the cerebellum to the cerebrum, from the cerebrum down to the brainstem. So you can really think of the thalamus as a relay center. The hypothalamus, which sits just below the thalamus, hence hypo, below, is our homeostasis regulator. All of our kind of whether it's body temperature, stress responses, water balance, hunger, thirst, all of those kinds of kind of homeostatic responses. The control center for all of those feedback mechanisms is the hypothalamus. And then lastly, we have the epithalamus, or really the pineal gland that sits in the epithalamus. The pineal gland, as part of the endocrine system, secretes a hormone called melatonin, helps put you to sleep. If we look then at the brainstem, we're connecting the upper and lower regions, right? So we're going to connect the cerebrum and diencephalon to the spinal cord. And then there are three parts of the brainstem. The midbrain is the kind of most superior portion, and then underneath that is the pons, and then inferior to that is the medulla oblongata, or the medulla. So again, think of the brainstem as our primitive brain, right? It's what keeps our body functioning, even if we have kind of no conscious sensation or conscious thought. There are a couple parts of the different parts of the brainstem. The corpora quadrigemina, for instance, which consists of the 2 superior colliculi and 2 inferior colliculi, do startle reflexes. So like when you like somebody like throws a hand up in their your face and you like pull back, that's a startle reflex. Or when you hear a loud sound, that's a startle reflex. And it's the corpora quadrigemina that helps you kind of process some of that. You also have some pain suppression centers in the midbrain, allows you to kind of ignore pain and keep on powering through, as it were. In the pons, you'll find 2 cerebellar peduncles that connect the cerebellum to the cerebrum and the cerebrum to the cerebellum, so we can get communication going there. You'll also find some control centers that particularly help us regulate our respiratory rates, particularly actually when we're exercising, when we're sleeping, and when we're talking. In the medulla, this is all where what we call the pyramidal motor tracts, so these big voluntary motor neurons, right? They're going to cross over. Right brain controls left body, left brain controls right body. That crossover for those motor neurons happens here in the medulla oblongata. And then you can kind of think of the medulla as, I don't know, but like a little worker bee for the hypothalamus, right? So the hypothalamus is the control center for all these homeostatic mechanisms, but the medulla then kind of takes the instructions from the hypothalamus and sends it out to actually the effector organs, so that we can regulate our heart rate and our blood pressure and our breathing, coughing, hiccuping, sneezing reflexes. It's all these brainstem nuclei that will help us kind of carry all that stuff out. So let's look at the diencephalon. You can see here is the thalamus with the hypothalamus below it. So, and then the pineal gland back here, very tiny little structure. So the thalamus is the relay. The hypothalamus is the kind of homeostasis center, and then the pineal gland back here for the melatonin hormone that helps put us to sleep. You can see here is the mammillary body, and then the pituitary gland. So again, mammillary body helps link smell and memory. The pituitary gland is a portion of the endocrine system, and you can also see like a cross section through the optic chiasm. It's where some of our optic nerves cross over in order to help us process our eyesight. If we look at the portions of our brainstem, the midbrain here with the corpora quadrigemina on the back, the pons here, and then the medulla oblongata here, then leading into the spinal cord. The last kind of major region of the brain is the cerebellum. It is very similar to the cerebrum in that it has an outer cortex of gray matter, then we find all the cell bodies, and then kind of inner white matter. And actually the inner white matter, I'm going to go back for a second. You can see it looks sort of tree-like, right? Here's the branch of the tree, and here's all the kind of branches, right? There's the trunk. A trunk of tree, all of the branches, and so very often the white matter of the cerebellum is referred to as the arbor vitae. Arbor means tree. In terms of what our cerebellum does for us, it does process a lot of sensory information, particularly about balance and body position, kind of like some muscle and joint stretch, like where are we in the world, and then it's going to help us kind of take that body stretch, body position information and equilibrium, and kind of send it to the prefrontal cortex, or excuse me, the premotor cortex, so that we can kind of coordinate our movement, right? Because we can't really get from point A to point B unless we kind of know where we start. Where are we at point A? And that knowing where we are at point A is very much a function of the cerebellum. We also have 2 kind of functional brain systems where they're not like just a single part of the brain. They're not just in the cerebrum. They're not just in the diencephalon. They're kind of found through kind of wide tracts of the brain. And those 2 systems are the limbic system and what we call the reticular activating system, or the reticular formation. So the limbic system consists of things like the amygdala, which helps us process our emotions, the hippocampus, which helps process our memories, the thalamus and hypothalamus, prefrontal cortex as well. But the limbic system is our emotional brain, our affective brain. When you're feeling emotional, it is typically the limbic system parts of your brain that are active. And then the reticular formation is a whole set of nuclei within the brainstem that basically take information from all of your kind of sensory input, right? Whether that's temperature and pain receptors, visual reception, auditory reception. It all kind of feeds in to the reticular formation, and then the reticular formation sends that information through the thalamus out to the cerebral cortex, and it basically helps keep your cerebral cortex awake and conscious and alert. It also kind of filters out some of the repetitive stimuli so that we don't kind of overwhelm the cerebral cortex. We want the cerebral cortex conscious, but we don't want it like just awash in sensory data so much so that it can't make sense of it. If we then look at the ventricles in the brain, so this seems somewhat counterintuitive, right? They're actually quote-unquote empty spaces in your brain. They're not empty. They're actually filled with what we call cerebrospinal fluid, and they also then connect to the central canal of your spinal cord. So there are 4 ventricles in the brain. You have 2 lateral ventricles that are in the cerebrum that are separated by this membrane called the septum pellucidum. The 3rd ventricle is enclosed around the diencephalon, and then the 4th ventricle sits between the brainstem and the cerebellum. So let's take a look, right? So here's your 2 lateral ventricles separated by the septum pellucidum, the 3rd ventricle in the diencephalon, and then the 4th ventricle between the brainstem and the cerebellum. And so essentially you have these kind of fluid-filled spaces in your brain. So it's not white matter, it's not gray matter, it's basically an empty space that gets filled with CSF, which that seems like kind of counterintuitive, right? Like, why not pack more neurons in there? But the ventricles and the CSF serve a purpose. And we're going to get to that purpose here in just a little bit. But because the ventricles are also connected to one of our kind of protective features for our brain, I want to talk about them first. And these are known as the meninges. There are 3 of them, and the dura mater is the outermost membrane. It's very leathery. The arachnoid mater is somewhat spiderwebby and kind of meshy, hence arachnoid, right, spider. And then the pia mater is this very thin membrane that actually will cling to your brain tissue. So the purpose of the meninges is to protect your brain. It helps separate the brain. It helps keep stuff from getting into the brain. But sometimes they can become inflamed and infected. And if you've ever known anybody with meningitis, that is inflammation of these meninges. So the outermost meninx is the dura mater. Just underneath that is then the subdural space, which gets filled with CSF. Just underneath that then is the arachnoid mater, which is kind of the middle membrane. And then just deep to that is then the subarachnoid space. And then lastly, the innermost meninx, the pia mater. And mater is a Latin word for mother. So dura mater, like durable mother, right? Literally tough mother. And so the outermost meninx, the dura mater, is this very, very leathery coating, whereas the pia mater is very kind of fragile and very well vascularized. So you can see here, of course, is the bones of the skull, right? That's another really good protective feature. The outermost dura mater, it's actually 2 layers, which is another reason why it's like super protective. And then the subdural space underneath that, and then the arachnoid mater with the subarachnoid space underneath that, and then lastly the pia mater, which is very, very, very well vascularized. So these kind of submeningeal spaces, both the subdural space and the subarachnoid space, like the ventricles, also fill with CSF. So you can see here's the CSF in the submeningeal spaces, and here it is in the ventricles, and then also traveling down the central canal of the spinal cord, because the ventricles are actually continuous with the central canal of the spinal cord. The structure that produces CSF is this very kind of vascular structure called the choroid plexus. And when you pull it out of the sheep brain, it looks sort of I don't know, fluffy is the word that comes to mind. CSF is very similar in composition to our blood plasma. So it's got all kinds of ions and nutrients in it. And that is the advantage to the ventricles. Right? These fluid-filled spaces in the brain essentially allow these spaces, the CSF, to fill these spaces, and it acts like a cushion around the brain in the submeningeal spaces, and it also lightens the weight of the brain. Your brain would be like 90% heavier than it currently is if you didn't have the submeningeal and ventricle spaces filled with CSF. The choroid plexuses contain ependymal cells. The central canal of the spinal cord is also lined by ependymal cells, and that is what then kind of makes the CSF and allows it to circulate throughout all of these spaces. The last bit of chapter 17 looks at the cranial nerves. Now again, these are actually part of the peripheral nervous system. They're not part of the central nervous system, but since the kind of origin of most of them is on the brainstem, and then some a little bit here on the forebrain, they are often studied as part of the central nervous system, but again, they are part of the peripheral nervous system. You have 12 pairs of cranial nerves, one on each side, and they are named and numbered for whatever function they happen to have. So for instance, cranial nerve number 1 is the olfactory nerve, right? Olfaction, smell. Cranial nerve number 2 is the optic nerve, so you can see. And very often they will tell you kind of in their name essentially what they do. They also all have a Roman numeral, so the olfactory nerve is cranial nerve number 1, optic nerve is cranial nerve number 2, etc., etc. Most of these cranial nerves are mixed, meaning they're carrying both sensory and motor information. You have 3 that are completely sensory only, olfactory, optic, and the vestibulocochlear. And then you have 2 that are primarily motor, meaning they're going to carry maybe a little bit of sensory data, but they're mostly carrying motor data. In terms of what I will expect you to know for the cranial nerves, you need to be able to find the particular ones that are on your like sheep brain list, but you do need to be able to name and number them. So if I ask you what is the function of the accessory nerve, you got to be able to tell me what that is. Kind of where their target is, so again what they are going to. Are they going to innervate a particular thing? Are they going to take sensory information from a particular organ, etc., etc.? And then which of them are sensory, which of them are motor, which of them are mixed. And again, most of them are mixed. And there's a lovely little table in your book. It's table 17.2, lists all the different cranial nerves. So that is definitely a very good resource for you to study for in order to prepare for the cranial nerve portion of this exam. And that is the end of 17.